This talk is going to deal with Neo-Darwinism and 10 ways that science falsifies that framework. Darwin has had quite a legacy. Some have suggested that Darwin's book on the origin of species by means of natural selection has affected the outlook of the modern world second only to the Bible. So his ideas have had a profound effect on our world, and I would say all of us here in this room. So I'm to begin with going to just give an overview of what Darwin's thinking was all about. So what was the essence of Darwin's argument regarding the origin and history of life? Well, Darwin argued that just as humans apply selective breeding to develop improved varieties of animals and plants, an analogous process, one he called natural selection, operates in the natural realm to yield types of plants and animals better suited to their natural environments. Okay? Actually, he went quite a bit further than that. But let's look at some of the kinds of, some examples of artificial selection, human selection, human plant, well, here's animal breeding. So we see these various breeds of cattle, some that are bred to produce either very high fat milk, like in the case of the Jersey, or a large total quantity of milk, as in the Holstein. And then several different breeds, cattle, beef breeds. There's in Darwin's day, most of these breeds existed in Darwin's day. We also see very familiar with all the dog breeds, all the different varieties of dogs that have been produced as a result of selective breeding. And in Darwin's day, there was also, it was popular to breed pigeons and to find all the different, in some cases, very spectacular examples of that could be produced from pigeons. Apparently, Darwin convinced himself they all descend from the wild rock dove shown on the upper left there. So Darwin was familiar with all of these achievements, and most of the biologists of his day were as well. So just how does this process Darwin envisioned work? Well, in natural plant and animal populations, variations in physical characteristics provide some individuals, Darwin argued, in each population an advantage over the others. Individuals with the advantage tend to leave more offspring than others, Darwin argued. If the trait responsible for the advantage is heritable, if enough generations elapse, and if environmental conditions do not change, Darwin reasoned that most individuals in the population eventually ought to inherit and express the beneficial trait. I might just comment here that what we're talking about is adaptation, which does occur in nature. But anyway, Darwin went further than that. He further argued that the tendency for beneficial traits to become predominant is enhanced by the fact that most organisms produce many more offspring than can be supported by the resources available. Hence, in every generation, there's usually a competition or struggle, often a fierce one, for survival. Acknowledging his ignorance of the actual source of the variations, Darwin nevertheless speculated that there might be no limit to the amount of possible variation available for natural selection to exploit. Now, this is where he really got into the realm of speculation, where there was no basis on his part to believe, to argue that there was no limit to the amount of variation available. Darwin convinced himself and multitudes of others after him that his hypothesis involving tiny step-by-step changes over immense spans of time could account fully for the history of life on Earth, from the appearance of a first primitive common ancestor to all plants and animals, fossil and living, as well as human beings. So basically, Darwin did a major extrapolation from the processes he saw in human, plant, and animal breeding to nature, and imagined that there was no limit to how far what that process could accomplish. But there were a number of things that Darwin did not know. One was the astonishing complexity of all living things. The scale of most important structures in living organisms, for example, are some 500 times smaller than what he could see with an optical microscope. There was no way he could see what life was really like, certainly not at the molecular level. So if he had understood just the complexity of life, he would have realized that his theory simply could not cope with that kind of complexity. Secondly, he did not know the source of the variations that plant and animal breeders exploit. Third, he did not know the laws of inheritance. A contemporary, Gregor Mendel in Austria, working with pea plants, derived some of the basic laws of inheritance, but Darwin never apparently knew of those. Fourth, the fact that life specifications are in the form of linguistic instructions. Darwin had no inkling about that. So, nevertheless, discoveries in the early 20th century led to an updated version of Darwin's hypothesis. And this took place in the early 1940s and became known as Neo-Darwinism. The main features of Neo-Darwinism were the Gregor Mendel's laws of inheritance were folded in. Random genetic mutations were invoked as the source of inherited variation. Mutation was discovered in the first 2 decades of the 20th century, mainly in the fruit fly lab at Columbia University. What was his name? Something Hunt Morgan. What was his first name? Anyway, Morgan's lab there, they found that radiation and also certain chemicals could produce changes in the genetic makeup of fruit flies. So at that point, that had been a big problem in the latter decades of the 19th century with Darwin's ideas. It was realized that there really wasn't a source of variation, or if it existed, nobody knew what it could be. So this discovery appeared to come to the rescue, these random mutations. So that was folded into Neo-Darwinism. And thirdly, Darwin's concept of natural selection was retained. So those are the main elements of Neo-Darwinism. Now, no update to that framework has occurred in the last 75 years. So Neo-Darwinism, as it was formulated in the 1940s, is still the current officially accepted view by the professional biology community about what has driven the history of life on Earth and what explains the diversity of life on Earth. However, discoveries made since the 1950s now present such major challenges for the Neo-Darwinian hypothesis that it is utterly untenable from a scientific standpoint. So I want to consider tonight and tomorrow morning 10 of these major challenges, each of them fatal is in my assessment. Each one of these 10, any one of them would be fatal. But taken together, it makes Darwinism, Neo-Darwinism, simply a charade. So the first one I want to put up here follows on from the previous lecture, namely that life's DNA specifications are linguistic and non-material in their ultimate essence. As a purely materialist hypothesis, Neo-Darwinism offers no explanation for this crucial aspect of every living system, its linguistic specifications. So this seems to be just so obvious. I don't need to say much more based on my previous talk, but let's look at it a little more. Well, no, I'm going to go through the 10 here. Yeah, that's what I'm doing. Number 2 is that Neo-Darwinism corresponds to a trial and error search process for genetic improvements. Yet if every organism which had ever lived were to represent a separate trial in this trial and error process, there are not enough trials available to test the possible configurations of even one single small life protein. So there's not enough trials available in this grand trial and error process that Neo-Darwinism assumes to make any difference at all. I'll show that's what this point, and I'll try to make that. I'll quantify that. I'll give numbers. Third major problem is known as Haldane's dilemma. In the 1950s, J.B.S. Haldane, one of the main architects of Neo-Darwinism, he was one of the key architects, did some further calculations and found that for a new trait to replace an old one in a population, a huge number of individuals must perish without leaving offspring. This extreme cost of substitution, as Haldane referred to it, makes Neo-Darwinism too slow and ineffective to matter for most multicellular organisms. So Haldane realized this was a gigantic problem for the whole framework, and it still remains an unsolved problem, but has become a trade secret that population geneticists just don't talk about. So most of the other biologists don't even know about it. 4, natural selection cannot even detect, much less eliminate, most bad mutations. Most biologists are not fully aware of that. So natural selection results in genetic decline, not in genetic improvement. And this, my friend John Sanford has referred to as genetic entropy, this inevitable genetic decline that's taking place in every organism, because natural selection simply can't detect most of the bad mutations. 5, biological systems are irreducibly complex and therefore could not have come into being by, using Darwin's words, numerous successive slight modifications, as he imagined. So we'll look at that. There's a book out there on the book table by Michael Behe, already published more than 20 years ago on that topic, Darwin's Black Box, where he talks about this issue of irreducible complexity. Number 6, favorable mutations are so rare as to be non-existent relative to the bad ones. And one reason is that each nucleotide tends to be a part of multiple messages because they overlap the way they do. And you make a change in one, it may not be lethal in one of the messages, but almost certainly it'll have serious consequences in one of the other messages. We'll get into that later. I'll go into more detail later. 7, even if favorable mutations were to occur, the waiting time for even short strings of them to arise together is prohibitively long. And I'll show that just getting two, if you need two for a new capability, and just using just some extremely favorable assumptions, it takes like 100 million years for just two to arise together. So that's another fatal problem for Neo-Darwinism. 8 is the discovery of what are known as orphan genes, genes that are unique to a given genus or species. These are found in every species that's been examined so far. And these orphan genes are fatal to the proposition of common ancestry. It means that every kind of organism has to be a unique creation and not related in this tree of life concept that Darwin had.
Could you hold that for just a sec while I get it written down?
I'll go into detail on this one tomorrow morning. Okay, number 9, the fossil record contradicts the simple to complex and common ancestry picture of the history of life demanded by Neo-Darwinism. So Neo-Darwinism is contradicted by the fossil record in some of its key features. And 10, many diverse lines of evidence indicate that the Earth is young, thousands, not billions of years in age, so that the vast span of time Neo-Darwinism requires simply does not exist. So that's another fatal problem. Okay, before we discuss these challenges, let us first consider what Neo-Darwinism must explain in view of discoveries of modern molecular biology, discoveries made during the past 60 years. These discoveries have revealed a level of complexity that is nearly beyond human ability to comprehend. Living systems rely on a vast array of miniature machines, each typically consisting of a few protein molecules. These machines are so small that effectively every atom makes a difference in how they function. Now, I already showed in the last talk this little animation of kinesin walking on a microtubule. I showed the microtubules are very, very carefully designed, as you see here, have a very regular design, also made out of protein. Here you see, I didn't emphasize it, these transport vesicles containing chemical species that are needed elsewhere in the cell and are moved from place to place in these spherical lipid transport vesicles. And actually there are labels that correspond to UPS-like instructions on where each container is supposed to go within the cell. We saw this microtubule network. Now, I show this again to make the point that for energy, each step of this little robot requires one molecule of what is known as ATP, adenosine triphosphate. And this is a rendering illustration of what this molecule looks like. It's the primary energy-carrying molecule which powers most cellular processors in all living organisms, from single-cell bacteria to plants to animals. Everything relies on ATP. And its energy is released when this last phosphate unit is detached from the rest of the molecule. Amount of energy that seems to be in the range of what most cellular processes require is released when that phosphate unit is detached. Okay? So as my second example, I want to highlight the machine called ATP Synthase. It's a rotary machine that restores that original structure of ATP, that joins that phosphate, that third phosphate back and raises its energy again to create new ATP. And this little machine spins at about 10,000 RPM, and it's driven electrically. It's an electrical machine driven by a flow of protons. And again, this machine, ATP synthase, exists in every form of life, from the simplest bacteria to plants to animals, yeast, everything has ATP synthase in it. So this is a little diagram of this ATP synthase machine. These are embedded in a membrane, as I'll explain later, in units, little organelles in the cell called mitochondria. And again, it shows here at the bottom left, ADP, adenosine diphosphate, plus a phosphate unit gets catalyzed, connected to form ATP, which can be reused for energy. So this bottom part has catalytic function. This upper part rotates, and it's powered by protons flowing through. There's a proton gradient across this membrane, and the protons flow through this machine to cause it to turn. I'll talk about that a bit in the next slide, but many of our cells have hundreds of thousands, in a few cases up to 1 million of these machines in a single cell, especially the cells that require lots of energy, like our heart muscles, or even our skeletal muscles, our liver. They can have hundreds of thousands to millions of these little machines. The amount of ATP recycled per day in our bodies is approximately equal to our body weight. So if you weigh, say, 160 pounds, roughly 160 pounds of ATP gets recycled in your body every day. You have about on the order of 1.5 ounces, and that ATP gets recycled about once a minute throughout the day. And so this is a major aspect of your body's metabolism. And actually, one of the issues with aging is that this machinery, it starts to get broken, and it doesn't function quite up to the level as it did when you were younger. So that's the reason I take, what is it I take? I'm already, anyway, it'll come to me. It's supposed to prevent dementia too. It's what's in the skins of grapes. Resveratrol. That's right. It's resveratrol. And this product I take is natural resveratrol from grape skins, and the advertise based on experiments with lab animals. The ad says, live 25 years longer and feel 25 years, live 25% longer and feel 25 years younger. So it's basically, the lab research indicates that it helps maintain the mitochondrial machinery, keeps that mitochondrial machinery running longer. So anyway, whether it works or not, we'll see, I guess. Okay. So again, this is the ATP we're talking about, and these these little machines restore that, and where these little machines located, where they're embedded in this membrane inside a mitochondria. This is a cutaway view of a mitochondrion. We can have as many as 1,000 of these small organelles in some of our cells, and the gold dots correspond to those ATP synthase machines. This is another diagram. Here, each color signifies a different type of protein. So there are several different proteins that come together to form this machine. Each ball in this lower part represents an individual atom, and in all there are about 90,000 atoms in this single machine. And so there's a significant level of complexity in this particular machine. And again, it's in the simplest forms of life on our planet. So if you're trying to explain where the first kind of life, what the original, if you're a Darwinist, think you had some kind of original progenitor appear, somehow arise spontaneously. It's got to arise apparently with these kind of structures already present. It just boggles the mind. That's why this so-called origin of life problem, hardly anybody talks about it. And the amount of funding for research has gone down as people have realized just how impossible that is. So this is a little animation of ATP synthase. And it shows the ADP and phosphate coming in on the left, and these pink ATPs going out. I guess some of them going out on the left also. But this shows how this machine works. Again, it's critical for every form of life. So all this complex machinery, such as ATP synthase, is manufactured within the cell. Not only does it exist there, but it's also created. It's also manufactured there. So all the instructions for each of the proteins is encoded in the DNA. And then there's a then this DNA orchestrated, we won't even get into that, how this gets orchestrated, but it will open up and allow a certain segment of that DNA to be copied into RNA, an RNA molecule called messenger RNA. And then that's taken outside the nucleus and fed into a machine called a ribosome to manufacture a protein. So here we see this light blue strip here. That's the messenger RNA with its sequence of nucleotides on it. And then there's another type of molecule called a transfer RNA, the tRNA, that has the letters for just one codon on one end and the amino acid corresponding to that codon on the other end. And those come into the machine, have to match up with this tape that's being fed with the machine, and the proteins are then linked together and to produce this growing protein. So we have a machine that will manufacture any one of the roughly 100,000 different proteins we have in our bodies. So all of this manufacturing, is taking place constantly in our bodies to keep our bodies, to have our bodies repair itself, and anyway, just a staggering process. So that's what Neo-Darwinism has to explain today. It has to explain all of that machinery, all of that DNA specification. So does it do it? Well, I say it's not even worth talking about. It comes nowhere close. So the first major challenge that I mentioned was that these specifications are non-material and therefore beyond the reach of any material explanation. And prior to the mid-20th century, no one even suspected that the linguistic specifications would turn out to be linguistic. And if they're linguistic, therefore non-material. And we've already seen that these specifications indeed are linguistic. There's an alphabet, vocabulary, and grammatical rules there. And we looked at this dictionary, codon dictionary for the amino acids. So the discovery of the details of how DNA carries and transmits heritable genetic traits resulted in a huge challenge. That challenge, how to account for coded linguistic specifications in a purely materialist manner. And in the previous talk, we saw why that represents such a challenge. And we saw that language is non-material. We saw because meaning is non-material, language is also non-material. But we also saw that matter has no ability to assign meaning to symbols to generate language. And therefore, the genetic specifications logically demand a non-material explanation, which Neo-Darwinism, as a purely materialist hypothesis, simply cannot provide. And we further saw that random processes can't generate meaning-bearing symbolic sequences. And as I mentioned before, that previous slide is a random sequence of 800 characters. And it's not surprising that it is nothing but gibberish. And the reason was that the defining aspect of language is missing, namely assignment of meaning. Again, random processes never generate meaning-bearing strings. Moreover, random changes in text strings that carry meaning-bearing messages invariably transform those messages into gibberish. So we see here a big problem with that random mutation as a mechanism. Those random changes, based on what we know about language, based on what we know about linguistic sequences, those random changes are guaranteed to ultimately transform the meaning-bearing DNA sequences into gibberish. That's another way of understanding genetic entropy. This implies that Neo-Darwinism, the Neo-Darwinian expectation that random mutations can indeed lead to improvements in an organism's genetic specifications has no logical basis. So again, to reiterate, these specifications are linguistic, non-material, and they absolutely require a non-material explanation, and Neo-Darwinism doesn't qualify. Okay, so as I mentioned before, most of what I've just said so far is in this paper, A Linguistic Argument for God's Existence, which can be downloaded also from another website I'm on, logosresearchassociates.org, as well as globalflood.org. Okay, now let's consider the second of the 10 challenges. Neo-Darwinism corresponds to a trial and error search process for an improved combination of traits. Yet even one live protein is so complex that the number of possible trials during the entire history of life is far too small to matter in such a search process. The Neo-Darwinian hypothesis in claiming that simple organisms develop into more complex ones require that hundreds of thousands of new proteins with new functionalities somehow appear. But how? How can new proteins possibly arise? The Neo-Darwinian answer is that random mutation of the DNA code somehow transforms the specifications for an existing functional protein into new specifications for a new protein with a new functionality. But crucial to whether the protein has function or not is whether or not it folds correctly. So I'll talk about that in the next slide. So what comes out of a ribosome is simply a linear chain of amino acids. It's called the primary structure. That linear chain of amino acids is manufactured by the ribosome. Now, because the side chains on the amino acid typically carry charge, either a positive or negative charge, that leads to a tendency for these proteins to form more compact structures. One structure is what's called an alpha helix, where this linear chain starts winding up into a helical shape. Another secondary structure is what is called a beta sheet, where this linear chain starts to fold back and forth on itself to form a sheet of amino acids. Okay, but then the crucial step is forming this what's called the tertiary structure, where the alpha helices and beta sheets collapse to form a protein, what's called a protein fold, a large three-dimensional structure called a fold. So all functional proteins are folded, and so a crucial thing to occur is for the protein to fold. If it doesn't have the correct distribution of charge on these side chains of the amino acid, it simply will not fold. It simply will not fold into a functional structure. So the proteins that fold are extremely unlikely, and that's been known for several decades. But the statistics of it were not well known. In 2004, a researcher named Doug Axe published results on what is referred to a site-directed mutagenesis experiments. So he selectively mutated, changed the amino acid along a chain, a protein that was 150 amino acids long, and looked to see how it affected the folding of that protein. And he found that the ratio of stable folds with function to the whole set of possible amino acid sequences had a ratio of 1 to 10 to the 77th power. So it was very extremely tedious research, extremely careful, but he was able to get this kind of estimate on the probability that you would get a stable fold and a functional fold out of all the possible amino acid combinations. So to say what I just said with maybe a picture or two here, if there are 120 amino acids in the chain and there are 20 different amino acids possible, the number of possible combinations, configurations of a chain that long is 20 raised to the 150th power, or 10 raised to the 195th power. That's how many possibilities there are. But then he, with his experiments, was to show that the number of those that would fold is a tiny, tiny fraction of that. Only 1 in 10 to the 77th power out of that large number there actually would fold. Okay? So how likely is it that a random event with a low probability would ever occur in the history of life on Earth. To address that question in an intelligent manner, we need an estimate for how many trials we have available in this trial and error process. Now, for most people, a good analogy might be, I think a reasonable analogy is winning the lottery. We can evaluate this question using the analogy of a lottery. The odds of winning depend on the number of lottery tickets one can buy. I forget what this latest jackpot, which was over 400 million, they said that the odds of winning were 1 in 200 and something million. So, if you went out and bought yourself, say, 100 million lottery tickets, you would think you'd have a pretty good chance of winning. Okay? Of course, most people couldn't afford that, or probably wouldn't take that kind of risk, and because the payoff wouldn't be that big. Okay, well, so but obviously the odds of winning depend on the number of lottery tickets, in addition to the number of balls and the odds in the balls. We can think of each genetically distinct organism as a separate lottery ticket in this trial and error process. So if we want to use the analogy of a lottery, we are thinking about how many lottery tickets there are. Let's assume that each living organism is represents a lottery ticket. And that we can do that if we assume that each organism that's ever lived is genetically different, genetically distinct from all the other organisms. So if that's the case, you can't, okay. The number can be no greater than the total number of organisms that have ever lived over the history of our planet. So let us estimate what that number might be. Some researchers, Whitman et al., estimate the reproduction rate of all single-celled organisms on Earth today is about 2 to the 30th power organisms per year. These are mainly bacteria that live in the ocean. The largest reservoir of living things on Earth are the bacteria that live in the ocean. There's quite a few that live in the soil on the continents, but there are more that live in the ocean. They took all these into account, came up with this number of 1.7 to 10 to the 30th organisms live and die on the Earth today. So let us assume at least one mutation per individual, such that all organisms are genetically different. So let us be generous and assume a time span of 4.6 billion years. Let us add an extra factor of 10 to allow for greater numbers of organisms during certain times in the past. Let's say for various reasons, conditions might have been more favorable than today for organisms to live in the ocean. If we multiply these numbers together, we obtain an upper limit on the number of genetically distinct organisms that could have ever lived during Earth history, of 10 to the 41st organisms. Okay. Behe, in one of his later books, The Edge of Evolution, uses a similar number, 10 to the 40th. He probably just did not include that extra factor of 10 that I'm adding in for good measure. All right, this limit of 10 to the 41st genetically distinct organisms that could have ever existed on Earth is a generous upper limit on the total number of trials, or if you will, lottery tickets available. So note that this limit is not merely for a single new protein fold, but is the total available for the entirety of the many hundreds of thousands of new proteins that the Neo-Darwinian hypothesis requires. So this is all the lottery tickets available for evolution of all of life through all of life history. But we'll see that it's not even enough to make any difference for a single life protein. So we saw the probability of finding one functional life protein fold in an amino acid sequence only 150 units long is only 1 in 10 to the 77th power. So if we use all our lottery tickets on this one protein, all 10 to the 41st tries, the odds of success are only 1 in 10 to the 36th power. That's one chance in a trillion of succeeding for this single short protein. So that's close enough to zero, what physicists would consider to be a likelihood of zero, to call it zero. So it's just irrational to think that this kind of random process could ever identify a protein that will fold of that length. This number of trials is available is so pitifully small relative to the number of molecular configurations that are possible. A trial and error process is guaranteed to fail even for one small life protein. And in my view, it is therefore inexcusable for anyone, certainly a scientist, to claim that trial and error processes can discover the blueprint for even one biomolecule, much less hundreds of thousands of them. It's inexcusable. There's no rational basis whatever for a scientist, educated as most scientists are, to make that kind of claim. These molecules are incredibly special. There's simply no material process that can possibly arrive at their design. This should be so embarrassing to the scientific community that they would no longer allow this kind of charade to continue. And that's why I wanted to have in the advertisement of this talk tonight and tomorrow morning, I wanted to have fake science as part of the ad on the ad on the bridge, and also in the what's it called, the Daily Post. Yeah, but unfortunately, certain individuals in this audience, maybe it was God's wisdom, I don't know, overruled. I don't mean to be unkind, Jenny. Okay. Let me emphasize here that I'm not claiming that mutation doesn't occur. Indeed, it does occur. In general, it is causing severe and irreparable damage to the genetic makeup. I'm just saying that this kind of process simply has no hope of success. This issue, the lack of enough trials, is alone sufficient to falsify the entire Neo-Darwinian framework. For this reason alone, Neo-Darwinism does not deserve even to be associated with the word science, in my opinion. So let me see where we stand on time. I think I will end it here with this statement. Therefore, okay, that was my already. I think that was my, yeah. All right, okay, did I go backward? Yeah. All right, therefore, it's my own conclusion that there's but one possible explanation, rational explanation, for how the living systems around us came into existence, namely, nearly instantaneous supernatural creation by the God of the Bible. I mean, is there a rational alternative? If anybody has a rational alternative, I'm eager to hear what that alternative might be. But I'm persuaded that no one has any other rational alternative. I suspect that the leaders, the leaders in these national science organizations understand that that the only alternative to Neo-Darwinism is nearly instantaneous supernatural creation by the God of the Bible. And they simply cannot bring themselves to admit that publicly. So that's the reason. And I think, but on the other hand, I think for just having a few Christians stand up and make this case, make it plainly, I believe that they cannot keep the dam plugged with their finger much longer. The evidence is just so overwhelming. So anyway, the other 8 challenges we'll take up in the morning, and I can move through those fairly quickly. I think I can get through those in the hour I've allocated. So we'll end at this point. I want to thank you for your attention and your interest in these matters. I might mention that I have been teaching. We've been trying to develop a course for training Christian apologists, people who can defend the faith, especially in these areas of science at Southern California Seminary. And I've been teaching a course called Understanding Darwin. And 3 of the books that I use are on the book table. I'm using the book Darwin's Black Box by Michael Behe. I'm using the book Genetic Entropy by John Sanford. I'm using the book Darwin's Doubt by Stephen Meyer. And I would say for people in this community, scientists, believers who are scientists, it's not that hard for you to come up to speed on these arguments that I'll be sharing with you. That I've shared tonight, I'll be sharing tomorrow. So you might give some thought to seeing if in your, maybe in some of your spare time, see if God might be calling you to that kind of service for Him. So I'll put on that brief recruiting hat here, if some of you think that might be a possibility for you. So with that, I'll close. Let me close in prayer. I hope that doesn't offend anyone here, but I think it's appropriate to do that. Heavenly Father, thank you. Thank you for making these things so clear in our day. Thank you for giving us such clear, objective evidence of your reality. I pray you might be at work in our country, our world, that you'll allow these very amazing truths, these amazing facts to be more widely known. That you'll raise up people who are bold and can share these things without apology, and many might the light might come on for many people. So thank you for this opportunity. I pray you'll refresh us tonight, allow us to come back alert tomorrow morning, allow me to come back alert tomorrow morning, and just thank you for your grace to us in Jesus. In His name, amen. So, we still have a couple of minutes. I'll maybe take a couple of questions here, and then if some of you want to stay afterwards and ask further questions, we can do that. Any quick questions anyone might have?
How does Neo-Darwinism answer your language objection? How do they?
As far as I know, no one, they haven't even heard that argument. I'm not aware of anyone even hearing that argument. On the internet, I don't know of a single ardent evolutionist even addressing this topic. And you know, they, in my opinion, they can't. They have no answer to it. I mean, some of them might try to deny that language is non-material. That's one reaction I've had when I've given my language talk. They simply refuse to believe there's anything non-material, despite all these lines of evidence that I present. Okay, well, thank you very much. We'll see you at 9 o'clock tomorrow morning.